You can imagine shear stresses on that. The thing
Rupture.
ruptures, and gee whiz, you think about some of the needles or aerosolizing these vaccines. What effect does that have on the solid lipid nanoparticle? But even more, imagine mRNA.
Let's go. Hey Nick, we've spoken about shear stress before.
Yes.
And I was wondering if you maybe can elaborate on how we know that shear is bad and how you quantify something like that.
Yeah, that's great. I know we do talk about shear all the time, particularly when we talk about delicate drug delivery like cell and gene therapies. We're delivering viable cells in some cases, and shear is a bad word in those situations. And there's a couple of ways that we can measure that. One of them is kind of in the design phase, if you will. Shear is a mathematical equation coming way back, I think, from Newton. And anything with math we can put into a computer, and there's these computer models. It's called computational fluid dynamics, and we actually have a whole group here that does computational fluid dynamics. And so you can model, the part and see where their eddies and things will form in the flow path. And that's a bad place for shear.
So you see that light up on the screen.
You can see it light up on the screen, you can understand it and design around that. But as I often tell those guys, and they kind of hate me for this, is garbage in, garbage out, right? And so if the model is based on some data that is incorrect, it only takes you so far. So you want to have good data. And as a biologist, where I like to do that is I like to run experiments and see where that comes from. And so we're talking about cell and gene therapies. I will often, particularly if we're testing a new cannula to deliver stem cells, for example, actually run stem cells in the cannula and run simple viability assays. And those can be done simply looking at the viability of the cell under the microscope, seeing any sort of effects there. You can have markers in there, such as green fluorescent protein, which we've talked about before, and seeing if the signal comes out, or even simple viability assays where you're looking at if the cell's transpiring, you can see certain signals show up in a microtiter plate. So those are all good. And also too, one of the things that we've done a little bit of work on, which is interesting, is not just viable cells, but mRNA. So we've had mRNA vaccines, all of us. And there are different strategies to making sure that those mRNA vaccines are capable and protected. And they use solid lipid nanoparticles. And so it's a lipid body, and you can imagine shear stresses on that. The thing ruptures, and gee whiz, you think about some of the needles or aerosolizing these vaccines. What effect does that have on the solid lipid nanoparticle? But even more, imagine mRNA, and it gets exposed now to the environment or shear itself, and then what effects are there? And there's a whole number of different things that we can do to look at that. We set up an experiment where you run the solid lipid nanoparticle with the mRNA in that. You can look at viability of the mRNA either through a transfection assay, look and see if the mRNA actually gets incorporated into a cell and does it do what it's supposed to do. Or you can look at the particles themselves and look at things like Zeta potential, which is, of course, if the cell gets ruptured, the lipids get exposed, and you have a different pH, if you will, and then so that changes, and you go, oh, there's a change in that Zeta potential, therefore there's some shear, and that's what you look at. So there's a number of different tools, really depending on what you're looking at, but that is how we understand if shear is an effect or not.
Yeah, well, that's very helpful because by testing it functionally, you really know what's going to go and happens in real life as opposed to a simulation only approach.
Absolutely, and not to discount the simulation, guys. That work is fantastic and critical, but it's always good to back it up with real world data and building your model based around that.
Great, thank you, Nick. That was very informative.
Have a great day.